lack operon. There are two types of genes in our body. One is called housekeeping gene, and the other is luxury. Housekeeping genes are those whose gene products are required all the time. Example, glycolytic genes. That means the genes that produce glycolytic enzyme. Luxury works on and off basis. That means whenever the gene product is required, these genes would be switched on. Whenever not required, they will be switched off. Now, one such luxury gene example is that of lack operon. Lack operon. Operon is a group of gene. They have a regulatory part and a structural gene part. So, these are the components of the operon. These are the three structural genes. This is lac I, promoter, operator. Lac I is the regulatory part, which would decide when the structural gene has to express itself and when it needs to be switched off. In case of bacteria, this is something that is seen in bacteria. So in case of prokaryotes, many genes give rise to one RNA. So they are polycistronic. So all these genes would make one RNA, and from there multiple enzymes would be produced. Lac Z produces beta galactosidase. Lac Y produces permease. Lac A produces trans-acetylase. Beta galactosidase breaks lactose into glucose and galactose. Lacoperon is the operon that works for lactose metabolism. So if the bacteria requires glucose or galactose or as a source of energy in itself, and it has lactose in its media, then these three genes would be expressed, which would help in breaking down lactose, absorption and utilization in the body. Permease allows, or it helps to take up lactose so that beta galactosidase can work upon it. Trans-acetylase helps in absorption of the same. Now, first condition when lactose is absent in the medium. Lac I is a constitutive gene, so it would express itself. Housekeeping genes are also constitutive. It would express itself no matter what. So lac I would produce mRNA that would produce repressor protein. Repressor has affinity for operator. It would go and bind to operator. So if operator region is blocked, if at all RNA polymerase binds to promoter, it cannot reach structural gene and no transcription and translation will take place. Such a system is called switched off. So if lactose is not there in the media or in the surroundings of the bacteria, why would it produce enzymes that helps in uptake of lactose or breakdown of lactose? There is no requirement. Now, another case, lactose is present. Same operon. Here, mRNA will be produced, repressor will be produced, but since lactose is now present in the medium, it will act as inducer, bind to repressor and make it inactive. Now this inactive repressor cannot bind to operator. RNA polymerase would bind to promoter, continue till this, produce these enzymes that helps in take up of lactose and its breakdown and absorption. So this is a switched on case. So when the operon is switched off with the help of lac I, that is called negative regulation, and this is positive case. Now, since the bacteria doesn't know when it would have lactose in the medium or when it won't have lactose, so some amount of permease is always required in the cell. So some amount of little bit of positive production of this protein continues. This is all about lac operon.